or LPT. So our group is assigned to discuss the fourth lesson on chapter 3, which is the aspect of gene therapy. So our lesson objectives: first, describe gene therapy and its various forms. Second, assess the issue's potential benefits and detriments to global health. So genetics in human diseases, there are about 4,000 human diseases are thought to be inherited. So scientists are making good progress figuring out where genes are located on chromosomes. So genetic diseases are caused by mutations or incorrect sequences in the normal form of the gene. But before we dig deeper about our lesson, let us first describe the word gene and the word therapy. So the word gene is the functional and physical unit of heredity passed from parents to offspring, and these are also pieces of DNA. On the other hand, therapy is a treatment intended to relieve or heal a disorder. So to combine both a gene and therapy, it is an experimental treatment that involves introducing genetic materials into a person's cell to fight or prevent some diseases. So it also a treatment or cure for disorders which is caused by a mutated genes. Let us now proceed to the basic processes of gene therapy. So there are three basic processes of gene therapy. First, replacing a mutated gene that causes disease with a healthy copy of the gene. Second would be inactivating or knocking out a mutated gene that is functioning improperly. Third, introducing new gene into the body to help fight against some diseases. So in general, a gene cannot be directly inserted into a person's cell because it must be delivered to the cell using a carrier which is called a vector. So what is a vector? A vector is a living agent that carries and transmits an infectious pathogen to another living organism. So there is this what we call as the vector system, and it can be divided into two. First would be the viral vectors, second is non-viral vectors. So first is viral vectors. It is a tool that commonly used by some biologists in order to deliver genetic materials into the cell. Second is non-viral vectors. These are the gene delivery vehicles that are based on delivering foreign genetic material into the cell by using inorganic particles like lipid-based vectors and polymer-based vectors, and it is administered by direct administration.
So in most cases, genetic material or gene editing tools that are inserted directly into a cell do not function. Instead, a vector is genetically engineered to transport and deliver the material. Because they can deliver the material by infecting the cell, certain viruses are used as vectors. When the viruses are used in humans, they are modified so that they do not cause disease. Some viruses, such as retroviruses, integrate their genetic material, including the new gene, into a human cell chromosome. Other viruses, such as adenoviruses, insert their DNA into the cell nucleus but do not integrate the DNA into a chromosome. Viruses can also deliver gene editing tools to the cell's nucleus. Virtually all cells in the human body contain genes, making them potential targets for gene therapy. However, these cells can be divided into two major categories. First is the somatic cells, or most cells of the body. Somatic cells do not reproduce because it affects only the targeted cells in the patient and is not passed down to future generations. Somatic cell therapy is considered a more conservative, safer approach. In other words, the therapeutic effect is limited to the person who receives the therapy. However, this type of therapy has its own set of issues. Somatic cell therapy's effects are frequent transient because most tissue cells eventually die and are replaced by new cells repeated treatments over the course of a person's life are required to maintain a therapeutic effect. To summarize, the results of any somatic gene therapy are limited to the patient and are not passed down to his or her children. And second is the cells of the germline or eggs or sperm. Gene therapy using germline cells produces long-term changes that are passed down to future generations. Gene transfer could occur in all cells of the developing embryo if done early in embryologic development, such as during pre-implantation diagnosis and in vitro fertilization. The allure of germline gene therapy stems from the possibility of providing a permanent therapeutic effect to all who inherit the target gene. Successful germline therapies open up the possibility of eradicating certain diseases from a specific family and eventually from the entire population. This, however, is fraught with controversy. So how is it done? In gene therapy, a normal gene is inserted into the genome to replace an abnormal gene that causes a disease. One of the most significant challenges in the process is the difficulty in releasing the gene into the stem cell. To release the gene, a molecular carrier known as vector is used, which must be very specific, efficient in the release of one or more genes of the sizes required for clinical applications, not to be recognized by the immune system, and purified in large quantities and high concentrations so that it can be produced and made available on large scale. Once inserted into the patient, the vector cannot cause allergic reactions or inflammation. Instead, it should enhance more functions, correct deficiencies, or inhibit deleterious activities. It should also be safe not only for the patient, but also for the environment and the professionals who work with it. Finally, the vector should be able to express the gene for the duration of the patient's life.
Types of gene therapy: somatic cell therapy, germline gene therapy. Somatic gene therapy involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. Somatic cell body cell gene therapy is a daily only the transfer of genes to the affected cell. Germline gene therapy involves the genetic modification of germ cell or the origin cell that will pass the genes on the next generation. Germline gene therapy will be the permanent transfer of a gene into the sperm or egg cell. Future generation will be cured. Stem cell gene therapy. Stem cells are mother cells that have the potential to become any type of cell in the body. One of the main characteristics of stem cell is their ability to self-renew or multiply, maintaining the potential to develop into the types of cell. Stem cell can become cell of the blood, heart, bone, skin, muscle, brain, among others. Stem cell gene therapy. Stem cells are derived from different sources, two of which are embryonic stem cells, somatic stem cells. Embryonic stem cells derived from a 4 or 5 old human embryo that is the blastocyst phase of developed The embryos are usually extras that have been created in IVF, in vitro fertilization clinics, where several eggs are fertilized in the test tube, then implant into a woman.
So when we talk about somatic cell, it is cells that contains of two chromosomes which is inherited from each parent. And it is also cells after the embryonic development. The examples of the embryonic development is the fetus. So what are the roles of the somatic cells in our body? So the roles of the somatic cells in our body is the cell is the responsible for the renewal, the growth and maintenance of the tissues and organs in our body. The organs that involved in somatic cells, for example, is in our heart and bone marrow and blood vessels and many other organs. Is gene therapy is good or bad? For me, I cannot tell if it is good or bad, but it is also good because it can cure illnesses like the blindness. It will slow the progression of the loss of vision, but it is also bad because of the some life-threatening, risky conditions that patient might experience from the therapy, like skin allergies, the damaging of the organs, and some certain cancers like the brain tumor. So the complications or the side effects is really bad. So this type of procedure is very risky, and this procedure is for those people who are risk taker only. Aside from it's very expensive, it is also dangerous because of the complications. Who decides which traits are normal about disabilities and disorder? I think for me are the medical doctors, because they are responsible for all the procedures that we will conduct to a patient if ever. So if ever we have a problem due to about health, so we're always going to the doctors and they will gonna tell us if what is the right things to do and how to solve the problems if ever. But there are some disabilities that also curable, just do the surgery, and the doctors also are the one who will conduct that procedure. So everything what we're gonna do about health and science, we're always ask the doctors if what are the best things to do. Will the high cost of the gene therapy is available only for wealthy? For me, yes, because I researched about it and it will cost millions of dollars. But there's some assumption also that no matter how wealthy a country, it still depends about the administrations if a patient will conduct that procedure, because it will take a long months or a year just to study about the procedure, and aside from it's very expensive, it is also risky and the procedure is very expensive. So I think it's only for wealthy. But if there are some organizations that support a patient for that kind of treatment, then much better if there is someone who will volunteer to help a patient just to undergo that procedure. Why the widespread of using gene therapy will make the people less accepted in the society? Actually, this topic is very controversial because aside from it's very dangerous, it will invite discrimination to those people with disabilities. And there's a lot of arguments and many people oppose about it because of religious grounds. Because some religions believe that our genes will represent who we are biologically. Should people will be allowed to use gene therapy to improve some basic human traits? For me, yes. Although this topic is very controversial, but actually this therapy is available only for those wealthy people. And if that person who wants to improve himself or herself during gene therapy, then as long as they can afford it, then it's fine. But the problem is it's just so very risky, and although it cures some illnesses, but there's some complications also. very serious complications like cancer. Gene therapy is one of the modern type of medicine that is available nowadays, and thanks to the modern technology, because it creates a lot of inventions that help a lot of people in terms of health. Gene therapy is also a therapy that is used to cure many illnesses. Example is blindness, and others use this also to improve their basic human traits. But there's a lot of ethical issues about it. There's a lot of issues that involves in this topic because it's very sensitive and very controversial. Many people argue because aside from it's very expensive, it is also life-threatening because of some complications. And many people also oppose because of some religious grounds, because they believe that our genes represents if who we are biologically.